Hey there everyone. In today's video we are going to study the properties of the synapses. In the previous video I'll do a quick summary on what all topics we had studied. So we saw the synaptic transmission, like we saw the types of synapses based on the part of neuron, based on the number of neuron, based on the nature of the transmission, nature of the transmitter. Then we saw the functional anatomy of the synapse, in which we saw the postsynaptic, presynaptic and the synaptic cleft. Then we saw what all it contains, and then the steps of the synaptic transmission in the form of flowchart, then the role of the membrane protein, the presynaptic mechanism, postsynaptic mechanism, and then we saw the role of the dendrites in the transmission and transmission through the electrical synapses, and what is the difference between the electrical transmission and the chemical and electrical transmission. So we completed all of this in the previous video in hardly I think 33 minutes. So that is worth it, and I have completed each and every single line of the textbook, so make sure to watch that. And I have made it in the form of flowchart so that it will be easy for us to understand and also get more marks in the examination. So now, talking about the properties of the synapses, there are five properties of the synapses. Okay, so first one is the law of forward conduction. Okay, what is the law of the forward conduction? Impulses travel in the forward direction. Okay, always the impulses that travel in the forward direction from the presynaptic through the synaptic cleft to the postsynaptic. Okay, in a forward direction from presynaptic to postsynaptic, as I already told, unidirectional. Okay, they are unidirectional. This is in the case of chemical transmission. Okay, the exception for this law is the electrical synaptic transmission, because it is bidirectional. Okay, it is bidirectional. So this is the law of the forward conduction. It holds good for the chemical transmission but doesn't hold good for the electrical synaptic transmission. Okay, now coming to the second law, it is the law of convergence and divergence. Okay, convergence, what does convergence means so many things or so many particles coming at one particular point is called convergence. Okay, so see, many to one projection, many presynaptic neuron project on one postsynaptic neuron. Many presynaptic neuron project on only one postsynaptic neuron causes convergence. So this is called the conversion. Now the total opposite is the divergence. One to many projection. Okay, one to many. One presynaptic neuron projects on many postsynaptic neuron. Okay, and this is basically seen in the sympathetic, and this is commonly seen in the CNS functions and stuff. So see, this is whatever you see here, this is the convergence and this is the divergence. Okay, convergence and divergence. See, convergence, it was many to one. See, there are three presynaptic neurons and only one postsynaptic neuron. So see, it is getting converged, right? So whatever neurotransmitter it is there, it is getting converged. So this is the law of convergence. And for divergence, it is one to many. Okay, one to many. See, there is only one presynaptic neuron and three postsynaptic neurons, and the neurotransmitters are getting scattered to all of the three postsynaptic neurons. So this is getting diverted. So this is the law of diversion, and this is the law of convergence. Okay, convergence and divergence. So now we will see the third law, that is the synaptic delay. Okay, the synaptic delay, time required for impulse to be transmitted. Okay, whatever time that is required for the impulse to get transmitted from the presynaptic to the postsynaptic through the synaptic cleft, it is called the synaptic delay, and it is 0.5 milliseconds. Okay, 0.5 millisecond. Now what all occurs in this 0.5 millisecond? See, it is very fast. Okay, just imagine you're touching a hot pan or a hot utensil. So how quickly do you pull your hand back? How quickly do you withdraw your hand back? So you know what, so this it is due to this. Okay, the 0.5 millisecond, it is a very short duration. So it is due to influx of the calcium ion. See, what all happens in this 0.5 second is that it influx of the calcium ion occurs. इन द प्री नॉब में कैल्शियम आयन का इन्फ्लक्स होता है, जिससे वेसिकल रिलीज होता है एंड वो वेसिकल से न्यूरोट्रांसमीटर रिलीज होता है. इन द प्री साइनेप्टिक नॉब द कैल्शियम आयन्स गेट इन्फ्लक्स एंड देयर इज रिलीज ऑफ वेसिकल एंड द रिलीज ऑफ neurotransmitter causing the postsynaptic potential. The changes sorry, so the changes occur in the postsynaptic neuron, the ionic bases and stuff, movement of the ions, and either depolarization occurs or hyperpolarization occurs, either excitation or inhibition respectively. So that occurs and the response is seen. So this occurs only in 0.5 millisecond. What does it help? The synaptic delay, what does it help in? It helps in finding out finding out number of synapses. Number of synapses. Suppose the time taken for the transmission, the synaptic delay is suppose 3 seconds. 3 seconds. So see, 0.5 0.5 0.5 0.5 0.5 1 2 1 2 3. So how many synapses were there? 1 2 3 4 5 6. So there were 6 synapses. It helps in finding out the number of synapses. That may be monosynaptic, that is 0.5 milliseconds, disynaptic, again 1 millisecond, trisynaptic 1.5 milliseconds, and polysynaptic, as I have already discussed here. More, you can take the number of your own choice. Now the next law is the postsynaptic potential. As I have already discussed about this in the previous video, the postsynaptic potential or the synaptic potential basically. So it is of two types, and this is due to the movement of opening of the calcium ions. The potential is generated, the opening of the channels, the opening of the channels, influx, exflux, conduction of the ions, due to which there will be change in the postsynaptic membranes, and it causes two things: either depolarization occurs or hyperpolarization occurs. Hyperpolarization generates EPSP and depolarization generates EPSP, hyperpolarization generates IPSP. So EPSP is the excitatory postsynaptic potential and inhibitory postsynaptic potential. So it leads to excitation and it leads to inhibition. So this is the law, that is a postsynaptic potential that is due to the influx or the conduction of the ions. We have already seen in the previous video, you can check it out. We have seen the whole mechanism as well in the form of flowchart. So what is the next law? It is the synaptic inhibition. See, neurons are highly interlinked. The neurons, they are highly interlinked. One stimulus can generate widespread impulse to many neurons. See, suppose these are the neurons. These are the neurons. Suppose this is one to many is divergence, right? So this is one to many, and there is one stimulus given to it, and the neurotransmitters, it starts spreading, divergence occurs, and all of these neurons get excited, and suppose even these are having connection of neurons is so much, they are highly interlinked. One stimulus can generate widespread impulse to many neurons. See, one stimulus is causing these many neurons and these many neurons to get activated, or impulse is getting transmitted. So due to which explosion can occur, which might not be required. So synaptic inhibition is very much necessary in this case. Synaptic inhibition prevents this explosive situation. It helps in it prevents the unnecessary spreading of the impulses. I told unnecessary spreading of the impulses is stopped by which law? That is a synaptic law. So these are the laws. These are the laws of the synapses. There is a law of forward conduction, that it only goes unidirectionally. Exception is the electrical synapses, which goes bidirectionally. Law of conversion and divergence we saw by the diagram like this, many to one and one to many. And then we saw the synaptic delay, that is about 0.5 millisecond. That is the time required by the impulse. The time required by the time required for the impulse to get transmitted. And we saw the postsynaptic potential, it can be depolarization or hyperpolarization causing excitation and inhibition. And then we saw the synaptic inhibition. Okay, synaptic inhibition helps in preventing unnecessary spreading of the impulses. So these are the law that we saw. Now we are going to see what is direct and indirect inhibition. So what is direct inhibition? By the name itself it suggests direct inhibition occurs due to formation of IPSP. So if it is inhibition, obviously IPSP should be formed, the potential inhibitory postsynaptic potential should be formed. So see, if this is the neuron A and this is the neuron B, so this is the neurotransmitter. Okay, it is getting transmitted to the membrane of the postsynaptic neuron. So IPSP will occur. Okay, it is direct. See, the neurotransmitter from one neuron to presynaptic neuron to the postsynaptic neuron, it is getting transmitted, it is direct, so it is called as a direct inhibition. It is due to the release of the inhibitory neurotransmitter. Okay, the example is inhibitory descending fibers in the spinal cord. Okay, this is the example for the direct inhibition. Now what is about the indirect okay, inhibition? I don't know why my accent sounds very bad today. I'm not liking it at all. So suppose this is A neuron and this is B neuron, and it is releasing excitatory neurotransmitter, but there is one more neuron that is stimulating this A neuron to stop releasing or reduce the releasing of the excitatory neurotransmitter. So it is acting on the presynaptic neuron, not on the postsynaptic neuron directly. First it is acting on the presynaptic neuron to not release or to reduce the release of the neurotransmitter, due to which IPSP will be formed on the developed on the B neuron. So this is not directly, that it is not directly, another neuron is coming and doing all this. So this is called the indirect inhibition, and it occurs through three mechanisms that I have written in the form of flow chart. So what is the first mechanism? Action potential. Okay, the action potential. Okay, the activation of the presynaptic receptors. All of us know that the receptors are present on the postsynaptic neuron, not on the presynaptic. It is present on the presynaptic also, but it is not activated. So what happens is, first activation of the presynaptic receptor takes place. Now calcium, chloride conduction takes place, not calcium. Calcium helps in transmission of the neurotransmitter. Okay, the excitatory neurotransmitter. So calcium conductance takes place, due to which chloride conduction takes place, due to which the calcium channel closed. Okay, if the calcium channel is closed, we know that it is very much important to have abundance of calcium for the transmission of the synapses. So what it does, this chloride conductance jab hota hai, to the calcium channels it get closed, so less or no excitatory neurotransmitter is released, which causes inhibition. So this is this is the first mechanism. पहले तो activation होगा presynaptic receptor का, जिसकी वजह से the chloride conduction takes place and the calcium channels they get closed, and the influx of the calcium does not takes place. So there will be less or there will be no excitatory neurotransmitter release, so that causes inhibition. This is the first mechanism. Then we have activation of the presynaptic receptor again. So this is a very important step. Okay, it can be common to many other mechanisms as well. So potassium channels get opened. See what happens, slow conduction में क्या होता है, the potassium channels, if they are conductance of the potassium channels, if it is increased, so slow EPSP occurs. Okay, so see, the calcium channels open and efflux of the potassium ion takes place, due to which there will be less influx of the calcium ions. So if there are no calcium ion influx, then know that inhibition takes place. See, calcium ion is directly linked to excitation. Okay, calcium ion, if somewhere, see here also calcium ion also the influx of that was reduced, or the influx was not taking place. Here also influx is not taking place, so it is inhibition. So whatever it is there, the calcium ion, it has to not get influx. So these are the mechanisms. Okay, so activation occurs and the calcium channels open, efflux takes place, due to which there will be calcium ion influx will be decreased and inhibition occurs. And the last one is direct inhibition of transmitter release. In this one, the GABA, okay, it is it is taking place. Okay, it is important. Okay, the direct inhibition of the transmitter release. Okay, directly we are inhibiting the release of the transmitter, as we saw here. This is whatever you see here is the GABA. Okay, this is the GABA that is not letting the excitatory neurotransmitter to get excited, or it is completely diminishing it. So GABA, okay, there are two forms, this is GABA A and the GABA B. So inhibition by increase conduction of the chloride ions. The GABA B does inhibition by the increase in the potassium conduction. I told no, potassium conduction whenever occurs, so there will be slow EPSP. I have already discussed in the previous video, please go check that video out. Okay, and increased calcium conduction also leads to slow EPSP or total inhibition. Okay, now what happens is I'll tell you the ionic bases as well. See, sodium and calcium, they are good for excitation, and this potassium and calcium, they are bad for excitation. Okay, these lead to inhibition and this leads to excitation. So this is about the indirect inhibition. We saw the types of inhibition. Now again we'll see types of inhibition में feedback inhibition and feed forward inhibition. Okay, back and forward, feedback inhibition, feed forward inhibition. So one such good example, or one such important topic in this is the Renshaw cell inhibition. Okay, what is Renshaw cell inhibition? So see, suppose this is the neuron 1 and this is the neuron 2. So we are studying the feedback inhibition. So what happens is, on the neuron itself, on the presynaptic neuron itself, we have a cell. Okay, a cell that is releasing some chemical which inhibits this particular first neuron to stop releasing the neurotransmitter. Okay, see, this is also a type of indirect. Okay, this is not direct, but it is also a type of indirect, but this is Renshaw cell inhibition, that is a feedback inhibition, in which the cell is present on the presynaptic neuron itself and it is releasing some sort of chemical that is causing the first neuron to reduce the release of the neurotransmitter or completely inhibit. So what happens, this will not get excited. The action potential, the synaptic potential and the changes will not occur in the second neuron. So this is a form of inhibition which we call it as the Renshaw cell inhibition. This is an important inhibition. Okay, so see, inhibits discharge from the same neuron. See, it is present on the same neuron, it inhibits the discharge of the neurotransmitter from the same neuron. It can also inhibit the neighboring neuron, which is called as a lateral inhibition. Okay, so suppose this is the neuron and this is the Renshaw cell and it is releasing the chemicals, and also there is one more neuron which is close. I'm very bad at this drawing and all, especially for the diagrams. Okay, so see, it can also inhibit the neighboring neuron, that is called as a lateral, because it is beside it. Okay, just remember it that way. Okay, so this is called as a lateral inhibition. We saw the feedback inhibition. Now we are going to see the feed forward inhibition. So Purkinje cell output inhibition, it is also called as that. What happens in this is, from parallel fibers originating from the cerebellar cells. Okay, the parallel fibers of the cerebellar cells originating from the cerebellar cells, what happens is activation. These cells activate the Purkinje cells. Okay, there are Purkinje cells as well. They activate the Purkinje cells, and these Purkinje cells, they release the inhibitory neurotransmitter such as GABA. I told no, there are two types of GABA, GABAA and GABAB. This does inhibition by increased calcium conduction, chloride conduction, and this does inhibition by increased potassium conduction. So this is about it. So what happens is, the parallel fibers originating from the cerebellar cells, they activate the Purkinje cells, and the Purkinje cells in turn release the inhibitory neurotransmitter like GABA, okay, whichever, and the inhibition takes place. Okay, inhibition takes place. Mosi fibers parallel fiber pathway, this is called as a Mosi fiber parallel fiber pathway, and also the Purkinje cell output inhibition. So we are done with the feedback and the feed forward inhibition. Now we are going to see what is the synaptic I'm recording, no? What is the synaptic facilitation? So like suppose if I have paused the I have paused the recording and then I keep on explaining, and then at the end I find out that I didn't record only, then it will be such a thing. So synaptic facilitation, what is synaptic facilitation? It is also called we'll see the presynaptic facilitation first. Okay, only one is there, presynaptic facilitation. Okay, it is also called as presynaptic facilitation. So what does it mean? The release of more neurotransmitter, okay, which helps in transmitting the neurotransmitter to the postsynaptic neuron and causing conformational changes, and impulses are like action potential is generated and the response is seen. So this is the presynaptic facilitation. If more neurotransmitter is released, it is called as presynaptic facilitation, nothing but the synaptic facilitation. So how does it happen? Let's see. This calcium is very important. Remember, calcium is very important due to prolonged voltage gated calcium channel opening. Okay, see, if the we know that at the knob, the synaptic knob, we have the channels. Okay, we have the channels and we have the vesicles as well. So as the opening of the channel takes place, voltage gated channels, the calcium ion gets influxed, due to which the vesicles release the neurotransmitter, and the neurotransmitter is further transmitted on the पोस्ट साइनेप्टिक न्यूरॉन के उसके रिसेप्टर्स पे पहुंच जाता है, वो बाइंडिंग टेक्स प्लेस. सो व्हाट हैपेंस इन दिस इज इट इज ड्यू टू द प्रोलॉन्ग्ड वोल्टेज गेटेड channels, okay, which causes release of the vesicles by the docking and priming method. I already told this in the previous video, please check it out, it's very important. And which in turn releases the neurotransmitter such as serotonin, and facilitation increases. So it is nothing but presynaptic facilitation, release of more neurotransmitter. What is the mechanism? How does it happen? Prolonged opening of the calcium ion channel, okay, release of the vesicle and neurotransmitter, and facilitation occurs, like serotonin is one of it. So now we are done with the synaptic facilitation. Now thoda hi bacha hai, abhi zyada nahi bacha. Summation. What does summation means? Literally, okay, summation means adding up, okay, adding up of the synaptic potentials, okay, adding up of the synaptic. We saw the synaptic delay, right? It is 0.5 milliseconds. So what is its significance? It helps in finding out the number of synapses. So if suppose if there are 2 synapses, that means there are 4 it is transmitted in 2 milliseconds, so that means 4 delays have occurred, okay. So to add up the synaptic potential means summation. There are 2 types, the temporal summations and the spleitic summations, okay. Now we are going to study about the occlusion, okay. What does occlusion means? Occlusion, so see, this is the neuron A and this is the neuron B, and neuron A is releasing, okay, is having 5 okay oh my god, 5, and the neuron B is also having the 5 this thing, okay, the releasing transmitters, okay. So see, what happens is it is having 5, 1 2 3 4 5, and even this 1 2 3 4 5. So the net, we have to see what is the net will be 8, 1 2 3 4 5 6 7 8, okay. 2 are occluded, okay, 2 are occluded, and see, because they share the same area, so 2 are occluded, okay, they are coinciding, and this process occurs simultaneously. This is called as the occlusion, okay, synaptic occlusion. So this diagram is there in the textbook also, just read. There is like hardly 5 to 6 lines about it. Whatever I told, it is the same thing, but more in the bookish language, okay. What is habituation, sensitization, and the potentiation? Habituation, what happens? Synaptic transmission decreases. In sensitization as potentiation, the synaptic transmission increases, and even in this, the synaptic transmission increases. Now the mechanism behind this is repeated harmless stimulus. See, repeated harmless stimulus causes prolonged inactivation of calcium channels. See, if calcium channels are closed, what happens? The neurotransmitter will not be released and inhibition occurs. So less number of vesicles and less number of neurotransmitter leads to lesser response, okay. So this is habituation. Okay, an example to this will be, suppose you're eating biryani for the very first time and you feel very good after eating. You feel the taste, you're feeling so good. Suppose you started eating biryani like every day for breakfast, lunch, dinner, all the 3 times you're eating every single day for a month or 2 months, you're eating biryani, then you will get habituated to it and you will not like eating it again and again. So see, what happens is inactivation occurs. Inactivation of what? Inactivation of the taste occurs. Okay, so what happens in habituation? The repeated harmless stimulus for prolonged causes prolonged inactivation of the calcium channel, due to which there will be lesser number of vesicle, as in case of biryani, lesser excitation or the lesser taste or the lesser craving of biryani. So lesser number of vesicles and less number of neurotransmitter, so it will be less. So this is about the habituation. Now what is sensitization? Sensitization increases the synaptic transmission. It is due to the harmful stimulus. Okay, the calcium ion increases, calcium influx increases, and it increases the release of the neurotransmitter, causing the response to be increased. Okay, now harmful stimulus. Okay, harmful stimulus such as if a person is consuming a lot of alcohol, is intoxicating himself with the alcohol. So what happens is it is harmful, right? So what happens, his body has to generate some response that this specific thing for your body is not good. So what happens, the harmful stimulation whenever occurs is given, the calcium influx increases and the release of neurotransmitter also increases. So this is called as the sensitization. Now habituation, sensitization. Now potentiation. See, there are two types of potentiation. Even in this, the synaptic transmission also increases. We have the post tetanic potentiation and long term potentiation. Now what happens is the increase. See, remember this one thing, calcium ions influx makes more release of the vesicles and the neurotransmitters. So see, calcium ion is increasing and it in the presynaptic now, causing the neurotransmitter to increase. So this is called as a post-tetanic potentiation. Now what is long-term potentiation? The calcium ions, okay, it is increasing and causing increase of the neurotransmitter. What is the increase of the neurotransmitter release is called as presynaptic facilitation. We have already studied, no, presynaptic facilitation, and it is due to the N-methyl D-aspartate or the glutamate. Okay, by this is called the long term and this is called the post-tetanic potentiation. So this is about the chapter, still synaptic plasticity and done. Okay, what is synaptic? What do you mean by plasticity? Plasticity is a modification. Okay, modification or alteration. Okay, it is called as a plasticity. Synaptic plasticity we are going to study here. It is the ability to alter the functions of the neurons. Okay, function on how does it take and how does it learn, memorize and all, we'll see. Depends on the nature of the stimulus. Nature of the stimulus पे depend करता है. Strength and duration of the stimulus. Okay, if the duration is longer or if the duration is shorter, or the strength of it, the changes are memorized. Okay, the changes or whatever alteration takes place in the synapses, in the neurons, it is memorized and it helps in learning and it helps in retaining memory by learning, due to which it learns new things. Our neurons, they learn new things. My pen died, charge khatam ho gaya. Okay, so it helps in learning. I'm doing it with my fingers now, by the way. Okay, so suppose you're touching, again an example, so you're touching a hot pan and you withdraw your finger back or your hand back because it's too hot to touch. It's not safe, it is harmful. You get this, sensitization also, and see, it alters itself, the stimulus or the response, it gets altered and it remembers, it memorizes. So next time you see the same hot pan again, you won't touch it because your brain has learned that last time जब मैंने touch किया था तो it was very hot and I had to withdraw my hand, it was harmful for me. So that helps in memorizing and learning, processing and all this happens. So this is the synaptic plasticity. It is very important. It also helps in retaining the memory, whatever you read. So we all know this curve, the forgetting curve. So it is also related to the plasticity as well. Yeah, so now you're reading it for the first time next month you recall the same chapter, you won't be remembering the whole thing that you studied, but at least a little bit, the diagrammatic or the pictorial or whatever. So it is all about the plasticity. So the next chapter will be on neurotransmitter, the types of neurotransmitter, the inhibitory, the excitatory, etc. Make sure to watch all of this. Thank you so much.